here are several enzymes, or proteins, let's put them in order. DNA A, DNA B, DNA pol 1, DNA pol 3. If we were to put these in order in bacterial DNA replication, what would be? The first one that would land would be 1. Because DNA A is the one that binds to the origin to destabilize. The next one that comes is 2. What does DNA B do? It's the helicase that opens it up further. Followed by 4, followed by 3. So if I added a 0, which is primase, where would primase be? Where would primase fit here? It will be after 2. Because you can't have a polymerase working without a primase. It needs to have a primer. What does pol 1 do? Removes the primer, replaces it with DNA. Right? If ligase was thrown in, let's say ligase was 5, where would ligase be? Would be last as well. Right? Remember, when they're done, they end up as decatenated. So you have to have a topoisomerase open them up, and so that you can get 2 separate circles. So any questions about that part at the end? It wasn't on exam 3, it's not going to be on the final, which was to replicate or not to replicate. But again, be able to put these in order. Remember, it's, when they're replicating, what's the mode called? If it's circular, if you have a circular, it's called the theta or theta, because at the middle of the run, you see the great letter theta. Don't worry about the distinction between leading and lagging, etc. And then the replizome. This is not detailed. Oh, sorry. So any questions about this? Okay, so transcription, translation, which is chapter 8, real quick. You have to know the difference between transcription and translation. And remember, I went over 4 main types of enzymes. You might get a question right there. So for example, if you have an enzyme that is called DNA-dependent RNA polymerase, what is it also called? You have a DNA-dependent RNA polymerase. No, that's regular transcriptase, right? Because it's relying on DNA to make RNA. So that's your transcriptase. If it's relying on RNA to make DNA, that's reverse transcriptase. Okay? So the RNA polymerase, right, is the one we're focusing on, is actually the transcriptase. You have to know, again, what is it made up of, what is the difference between template and non-template strand, coding and non-coding. You don't have to worry about sequences of the promoter, but be able to, so for example, if the question is, the promoter of a gene is transcribed, true or false? The promoter of a gene is transcribed. False. The promoter of a gene is translated. But still false, right? If it can't be transcribed, it can't be translated. The promoter of a gene is replicated. True, it's got to be replicated because the cell needs to make copies of for daughter cells, right? Another point, the template strand for any gene goes 3 to 5, true or false? Template strand for any gene goes 3 to 5. And that's true. Why? Because DNA and RNA are made what? 5 to 3. So the template has to go anti-parallel, right? What is the makeup of the RNA pol holoenzyme in E. coli? Remember, you have the RNA pol holo. Holoenzyme means the entire. So what do you have? You have alpha 2, beta, beta prime, sigma. If it's this part, it's called the core. If it's this part, it's called sigma. If it's together, it's called the holoenzyme. What does it do? Real quick, it binds to the promoter, right? Opens up the DNA and starts transcribing. Right? At the end, it's going to hit a termination, transcription termination stop. There are some that need help, and the help comes in the form of which protein? Rho. So that could be a point right there. Sigma is used at the start, rho is used at the end. And it's easy. Sigma is start, SS, right? Terminator, the R is at the end. That's at least how I remember them when I was your age. Anyway, so questions about this. So that's transcription in a nutshell. But again, you might get a question where I give you the DNA and ask you to transcribe it. Remember, they go anti-parallel. So I might show you all of them 5'. So you have to be able to know that the 5' end comes at the 3' end, whatever. Okay. There's also types of RNA that we discussed. Please know them, right? True or false? The cell has only 3 types of RNA: M, transfer, and ribosome. Messenger, transfer, ribosome. That's big false, right? There is a big false. Because you have others. You have catalytic RNA, you have small RNA, you have tmRNA, right? So please remember these. Then before translation there was the genetic code. You'll get 1 or 2 questions on the code itself. So for example, first of all, remember there's 64 codons, right? How many of them code for an amino acid? 61. These are called sense. The nonsense or stop, there's 3. Please know them. UAG, UAA, UGA, right? It is degenerate. True, meaning what? Meaning there are more than 1 codon for a given amino acid. Okay? It is commaless, there's no stop. It is triplet, read in 3s, right? It is universal, true or false? It's not actually, right? Universal means every single person and organism has it. No, it is nearly universal because there are exceptions. Right? Then we go into translation. Remember the components. Again, I might not write it just to try to speed things up here. But for translation, in fact, let me give you a key here and ask you several questions about it because I will summarize a few things. Messenger, transfer, ribosome. If you guys, did you sign up the sheet before you leave? Did you? Just thanks. Okay, so messenger, transfer, ribosomal, 2 or all of the above. Which one of these is translated? If the question was this, which one of these is translated? A is the only one that's translated. Which one of these is transcribed? E, all. Which one of these is involved in translation? Involved. All. How? Messenger carries the message. Transfer brings the what? The amino acids. Ribosomal is the workbench. Right? So be careful what the question is asking. Now translation, just like I asked you for replication, you will be given steps, asked to put them in order. You will be asked questions about specific roles. So remember, there's 3 sites, EPA, right? There are 3 initiation factors. In fact, the only initiation factor you need to know is IF2. Because IF2 does what? Brings the initiator tRNA into the P site. Right? And in bacteria, what is that amino acid? The formal methine. In archaea, it's regular methine. So once it's in the P site, which one, the 30S, remember, comes first, the 50S joins later. You have a total 70S now, right? So once you have the 70S, the initiator is in the P site. The next one comes into the A site. What brings it? EFTU. Brings it in. What links the two? Peptidyltransferase, right? Which is it's a protein. True or false? Peptidyltransferase is the only one that's actually an RNA. It's a ribozyme, right? So, very quickly, IF2 does this, EFTU does this, peptidyltransferase does that, right? And then what does this? EFG. What is the role of EFTS to bring EFTU back into circulation? right?